Namaste. हमेशा खुश और खुश I'm Jagan Dr. Vinity and today I'm going to take a lecture on soybean diseases. Now, let's start with soybean disease. The first disease which we are going to take is soybean mosaic virus. The disease plants here, the symptoms are that the disease plants are usually stunted and distorted. That is, the leaf may pucker up, may get crinkled, or can be narrow or roughened up. That is, the smoothness of the leaf is not clear. Now, the infected seeds fail to germinate or they produce diseased seedlings. And the pathogen of this mosaic disease is a virus. It is flexuous particle. It's 750 to 900 nanometer long. Single-stranded RNA genome is there. The disease cycle is that it is a seed-borne transmitted through sap transmission and also through aphids. Now they are involved in the transmission. The favorable conditions are 18 degrees humid temperature and humidity is required. For management, we have followed the general practices like summer plowing, resistant varieties used, healthy and certified seeds. The disease, the field should be free from the weeds, and of course, we have to roll out the infected plants, that is, and burn them. Pre-sowing soil application of 4-8 at the 10 gram kilogram per hectare rate is also helpful. Two foliar sprays of thiamethoxam 25 Wg at the rate 100 grams per hectare or methyldimetron 800 ml per hectare at 30 and 45 days after sowing is also very helpful. The second disease is Rhizoctonia avian fly. Now here you see this is the infected seedlings and this is the infected plant. Now the causal organism is Rhizoctonia soli. The infected seeds have irregularly tan-shaped or light brown sunken lesions, and the leaves appear as water-soaked at first. Soon they take the reddish-brown appearance, and the infected portion turns tan-brown or black in color. Now, when there is high rainfall and humid conditions, a mycelial growth of fungus also forms in the leaves. Dark brown sclerotia forms on leaves and fruit. This pathogen survives as sclerotia in the soil, and the humid and cool weather is favorable for this disease. For the management, we have to avoid dense planting, completely cover the plant residue by clean plowing the field as soon after harvest. The infected stubble is destroyed and the seed treatment with iron and carbamazepine is used at the rate of 2:1 at the rate 3 grams per kg seed. Mancozeb or copper fungicide at the rate 2.5 gram per liter or carbendazim 1 gram per liter spray is also helpful. Third is bacterial plants. Pseudomonas syringae, Pachyerate, glyphene. Symptoms are the seeds may develop raised or sunken lesions and become discolored or shriveled. Small, angular, translucent, water-soaked, yellow to light brown spots appear on the leaves. The young leaves are almost infected, mostly infected and destroyed. They become stunted and florid. Angular lesions produce enlarge and finally merge to produce large areas which are dead. This causes early falling of leaves or defoliation of lower leaves. The large black lesions develop on the stem and leaves. Now you see the picture here. These are the lesions. Now how do we manage this disease? Deep summer plowing. Use of healthy certified seeds. Destroyed infected crop debris. Key treatment is streptocytosis. Why we are using streptocytosis? Because this is a bacterial disease. At the rate of 250 parts per million that is comes to 2.5 grams per 10 kg seed. Application of any copper fungicide at the rate 2 gram per liter along with tetracycline at the rate of 250 ppm is very high. Now, another disease and the last disease that is Pythium seedling and seed rot. So here you see, this is a diagram of the seedling picture, and you can see here the rotting is happening. So Pythium can attack and rot the seeds and seedlings prior to emergence and also cause the post-emergence damping off in wet conditions. Now, the most characteristic symptom is soft brownish colored rotting tissue. As you can see here, the rotting is taking place. And the secondary roots can be soft rotted, and therefore the plants will be stunted and killed in some cases. Now, how do we manage this disease? It is Pythium is very harmful. It affects all the stages of development, and it is active in wet soil. Now, it is a soil-borne fungal-like pathogen, and many different species attack soybean. So the host range is also very wide. So what we are going to do, soil drainage. Soil drainage is the most important disease management practice for seedling rot. Low areas of field should be avoided, and if there is already a history of seedling or root rot problem, then seed treatment of seedling rot can be seed treatments to reduce seed rot is taken. So students, I hope you have liked my lecture. If you have any queries, please fill in the comment section. This session was powered by digital version 2.0 of Digital Vidyapeeth University. Namaste.